Thank you, everybody, for joining us today for this webinar. Good afternoon. I am Yvonne Lucy. I'm one of the scientific curators at UniProt, and I will guide you through this webinar today on accessing human disease and variant information in UniProt knowledge base. I will take you through the website and a few examples, and will show you how you can explore the data we offer for human disease and variant information. In this webinar, you will learn how to navigate UniProt knowledge base to find and discover information on proteins involved in human disease and disease-associated protein variants. You will learn how to search for a specific disease or a protein variant, how to visualize how disease variants may affect protein structure and/or function, identify links to external disease-related resources, how to access, download natural variant information and data sets, and understand how UniProt annotates disease data and the clinical significance of human variants. But first, I will give you a short introduction to UniProt. So UniProt is the universal protein resource for comprehensive, high quality, and freely accessible protein sequence and functional information. We offer primary sequences, including sequences of isoforms, variants, and post-translational modifications, physiological protein functional information, including subcellular location, pathways, reactions, interactions, as well as involvement in disease. We provide structural information, including topology and access to 3D structures, and data analysis tools such as BLAST and multiple sequence alignment. One of the main activities our users perform on UniProt is to search for proteins and protein-related data. You can see here the different data sets. These are the data sets that you can search in UniProt. The one that is searched most commonly is UniProt knowledge base, or UniProtKB, which is the blue box on the left of the screen, and also the default data set for the search bar above. There are two types of protein entries in UniProt, SwissProt and Tremble. SwissProt contains all the entries that are manually annotated and reviewed by one of our curators, including sequence analysis and information from literature. The SwissProt entries are tagged with a gold icon, which means the entry is reviewed. The second section in the knowledge base is Tremble, and this section is automatically annotated based on rules and machine learning approaches that learn from the information in the SwissProt section, but is not reviewed by one of our curators. The Tremble entries are tagged with a gray unreviewed icon. There are a number of other data sets that can be searched in UniProt. These include species proteomes, protein clusters, and sequence archive. I would also like to draw your attention to the supporting data at the bottom of the main page, where you can find the human disease data set, which lists all the human diseases that are annotated in UniProt and the associated proteins that are involved in the disease. We will look at this in a bit more detail in the following slides. There are also 4 analysis tools that we offer on UniProt. We offer a BLAST sequence similarity search tool, a sequence alignment tool, and ID map tool, and a peptide search tool. So when would you come to UniProt for disease and variant information? Our users look up disease variants and their relationship with function and disease. And you can start by looking for information with a known disease, a known protein, or a known variant identifier. And I will show you now in the following slides how to search in these 3 different ways for your information. So in UniProt, you have different options available. You can search for a disease or protein in the free text search bar, where the default search is set to UniProtKB. You can also use the advanced search on the right-hand side, where you can combine search terms. Another way to find disease information and proteins that are associated with a disease is to search for the disease in the human disease data set that I mentioned before. This you can find under supporting data at the bottom of the UniProt main page. Here, all the human diseases are listed that are manually annotated in UniProt, including proteins that are associated with the disease. Currently, there are 6,322 human diseases annotated in UniProt as of February 23. You can then go ahead and choose your disease of interest from the list. Another way of finding your disease of interest is by going to the advanced search on the main UniProt page and select under pathology and biotech the disease in the drop-down menu, and then type in the disease of interest, for example, breast cancer. You will then be presented with a list of proteins that have been annotated having a direct involvement in breast cancer. To help choosing the protein we are interested in, we can edit the columns in our results table by clicking on the Customize Columns icon. Here, we can use the search box to look for columns related to human disease and natural protein variants. By searching for disease, you can add a column that shows the protein involvement in disease, as I have done here already. You see the column disease involvement and the proteins involvement in breast cancer. And then you can go ahead and look for variants, and you will be able to add a column exhibiting natural variants that are known for the protein. Now our table shows us additionally the disease and variant information available for all our results. So let's say you are particularly interested in the breast cancer type 1 BRCA1 protein and its involvement in breast cancer. We can either scroll down the results table to find the entry in the list, or we can search for the entry via the search bar under its gene or protein name. We can then access the entry via its protein accession number and go to the protein page to gain further insight into protein function and involvement in disease. On the protein entry main page, we find information on BRCA1, including the protein and gene name, the status, here a reviewed SwissProt entry, the organism, the amino acid length, protein existence, and the annotation score, with 5 out of 5, meaning there is a good amount of information available for this protein. You will find information on the molecular function of BRCA2 at the bottom here, and including references with the PubMed ID, from where the information is coming from that was curated in this entry. And you can find further information on the protein by clicking the tabs on the left-hand side. And we can now select the disease and variants tab to go to the disease section of the BRCA1 entry. Here we find the involvement in disease subsection, which provides the information on the role that the protein plays in breast cancer and links to the reference evidences, as you can see here with 22 publications describing its involvement in breast cancer. A note in this section is used to describe the role of the gene protein in disease pathogenesis and to distinguish, when possible, between causative, susceptibility, or modifier gene according to the literature and the online inheritance in men OMIM reports. In the case of BRCA1, the disease susceptibility is associated with variants affecting the BRCA1 gene. Yes, in the description field, you can find the description of the disease phenotype, which is mainly taken from the OMIM database. And you can also find the link to the OMIM database here in the description note, where you can find further information on the protein and the disease. Below the disease description, you will find a tabular view of any variants in BRCA1 that are related to the disease. Here we can find the unique variant ID given by UniProt, the nature of the amino acid change, the effect of the variation on the protein, the cell or the complete organism, and including, so here you find the disease association with here the acronym BC for breast cancer. You will find also an explanation of the variant pathogenicity. You will find a description of the molecular effects of the variant if they are described in literature, a link to the DB-SNP database, as well as literature publications that are describing the variant and its effects. So in UniProt, we are manually curating protein coding variants, including variants that are not linked to disease, which are mostly single nucleotide changes, and which can be linked with a polymorphism comment. We are curating disease-linked variants, which are mostly germline, not somatic changes involved in Mendelian disease and linked to phenotypes described in the OMIM database. We are also curating RNA editing events that lead to protein variation. The clinical significance of protein variants is assessed by our curators by the guidelines of the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. And we are currently distinguishing 3 pathogenicity classes in UniProt. The disease-causing variants, which are the likely pathogenic or pathogenic variants, variants of unknown pathological significance, and likely benign or benign variants. So going back to the BRCA1 entry, we can find cross-links to human-specific databases and other relevant resources towards the end of the disease and variant section. Here you can find cross-links to human-specific databases such as OpenTargets or Orphanet, links to genetic variation databases such as ClinGen and GenCC, and links to chemistry-related databases such as the drug compound resource Campbell. Following the link for Campbell brings us to the target report card in Campbell. So, and here you can find associated compounds, drug compounds for BRCA1. And by clicking on it, you can see that there are over 15,000 compounds for BRCA1 in the Campbell database. And following this link, you will be able to see them all listed either in a table view or the card view. And you can also filter for small molecules or proteins. Going back to the main page of UniProt, I will show you another way of looking for disease data. So let's search for another protein of interest, for example, EGR2 in the advanced search, where you can combine different search terms. So we can search, for example, for the gene name, EGR2, and restrict our search to human in the taxonomy search term. So this search will then give a list of sequences for the EGR2 protein that are stored in UniProt. You can choose the entry at the top with the gold label. This is the reviewed entry containing the manually curated information regarding the protein. The other entries at the bottom or below are the Tremble entries with the gray Tremble entry icon, and those are unreviewed sequence entries for EGR2. You can then open the entry page for EGR2 and go to the disease and variant section to look at the involvement of EGR2 in disease. Here you will find that EGR2 is involved in 3 different types of diseases, all with various variants leading to distinct disease phenotypes. Going back to the entry page of EGR2, you can select then the Feature Viewer tab at the top of the entry. This allows you to switch the view from the entry view to the feature view and will allow you to look at the protein features, including protein variants, in a more visual manner. The ProtVista feature viewer allows you to view the variants of your protein in the context of other protein sequence features. The features are listed along the protein sequence, as you can see here, and they are grouped into categories. You will find sequence features annotated, such as domains and sites, post-translational modifications, molecule processing, mutagenesis of different residues, as well as the variants at the bottom here. You can click on the variant tab to expand it further and see the variants displayed. So the variants view maps variants on a matrix with the sequence position on the x-axis and the amino acid changes on the y-axis. Here you will find manually curated variants as well as variants coming from large-scale sources. And so the large-scale variant data is imported from a variety of resources to complement the set of variants captured from the literature by our curators. And these resources include ClinVar, Cosmic, GNOMAD, and 1000 Genomes Project. You can filter the displayed variants by either the provenance, so you can choose only to view the UniProt reviewed variants or variants coming from large-scale studies, or ClinVar, and you can filter for the consequence of the variants. You can look at only disease-associated variants or benign variants in green, or then variants with predicted consequences. So let's filter for the pathogenic variants for EGR2 by pressing on the red filter for likely disease variants. So by looking at the pathogenic variants only, you can get a clear picture how the pathogenic variants are distributed along the protein sequence, and also then analyze the potential overlap with protein features that are shown above. You can then go ahead and select a feature of interest. For example, the zinc finger domain that is spanning the EGR2 residues 370 to 392. You will then, by pressing on this feature, you will then find a yellow bar highlighting the overlap with the protein sequence and the variants that are lying within this region of the protein. You can then click on a variant and find further information in the gray box, such as amino acid change, the consequence, and also the disease association. For this specific variant here, this leads to Charcot-Marie-Tooth disease type 1D, this variant that is lying in this zinc finger domain. So the third and last way to search for variant information is searching for a variant with its dbSNP identifier. So, for example, you have the dbSNP identifier for the presenilin 1 protein, which is RS63750001. You can take this dbSNP and go to the advanced search in UniProt and search for it under the search term genetic variation databases dbSNP. And you will then get the entry for the human presenilin 1, which contains this variant of interest. And let's go again to the feature viewer of this protein to look at the variant in more detail. So you can look up the variant in the ProtVista feature viewer, and you will find the variant here. It is a variant of residue 435, which is involved in Alzheimer's disease 3. And by clicking on the variant, you will get, again, a yellow bar that is highlighting eventual overlap with protein features. And following this highlight, you will find that the variant is overlapping a protein feature that is indicated here in the domains and sites. By clicking on this feature, you will find that this feature is described as palmotive, that is spanning residues 433 to 435. And this palmotive is described in the literature in this publication with this PubMed ID, and you can access this reference by PubMed or EuroPMC to read more about the palmotive in presenilin 1 and the putative effect of the variant lying in this region. Further, you can also analyze the impact of your variant, or where your variant is lying in the 3D structure of the protein, because the feature viewer also allows you to visualize 3D structures of proteins, both the experimental structures from PDB or predicted structures from AlphaFold. So you can select the source of the structure at the bottom here, either the experimentally deduced structures from PDB or structure predictions from AlphaFold. And you can also filter for the method that was used. So we are choosing a PDB structure, and you will find the structure displayed here in the window, and the variant that you have selected will be highlighted in green in the structure, allowing you to analyze where exactly this variant is located in the protein structure and possible consequences of this. So in the end, I would also like to point out that you can find further information about your human protein of interest in other species with orthologous proteins. So in our curation efforts, we curate orthologous proteins, which we identify by means of bioinformatic and functional analysis. And we group them in orthologous groups and protein families. And you can find those by blast search or family search, and look then at the overlapping features and conserved sites within the alignment tool, where you can highlight also mutagenesis assays or variants in the proteins of interest. You can then also find further information in the phenotypes and variant section of these proteins by looking at the disruption phenotype of knockout models or of mutagenesis assays of residues of interest. And in this way, you can learn more about your protein of interest. So in the end, I would like to draw your attention to the UniProt data that we offer. This can be found on the UniProt main page. Under the FTP download, you will find the list of human diseases that I mentioned, together with the associated proteins with these diseases. You will also find a list of human missense variants annotated in UniProt, as well as a file with variants imported from ensemble variation databases. This is for human and also other species, the one from ensemble. And under programmatic access, you are able to access information on variants imported from large-scale studies, via the proteins REST API, including genomic coordinates of UniProt sequences and variants of services providing annotation imported and mapped from large-scale data sources such as 1000 genomes, EXAC, and others. And you can find further documentation about the programmatic access here, and also help for programmatic access under uniprot.org/help/programmatic access. So in the end, I would like to acknowledge our funding bodies, the various institutes of the National Institutes of Health, the European Molecular Biology Laboratory, Open Targets, as well as the Swiss State Secretariat for Education, Research and Innovation.
Thank you, Yvonne. Thank you very much for today's webinar. And thanks everyone for joining in. And thanks for sending lots of questions, actually. So we have quite a few questions, Yvonne, for you. All right, so first question is, if you want all manually curated human variants, you may want to use the FTP file.
So under the FTP download, you find a list with manually curated variants. And these are missense variants that you can find a list there, and you can also see the association of those variants with the human disease that they are involved in.
Okay, thank you. Next question is, what are the links between the UniProt variant classification versus the CleanVar variant classification? Are they different or they are the same?
No, that's a good question. So there is a difference that CleanVar is using 5 categories. So this is ranging from benign, likely benign, unknown significance, likely pathogenic. And as I mentioned, our classification groups those variants into 3 groups. So we throw together the benign, likely benign ones and likely pathogenic ones. But we are following the same guidelines to classify these variants. And so the aim is in the future to have the same classification as CleanVar and distinguish the 5 different categories, actually. But yes, so this is work ongoing.
Okay, thank you. There was a question which came in chat. Where do you find variants in non-canonical ORFs encoding peptides or proteins?
So yes, we have the canonical sequence that is displayed, but we are also annotating variants that are not in the canonical sequence that are in the UniProt entry. And
there's another question about CleanVar. So CleanVar recommends submitters to use ACMG or AMP and CleanGen.
Yes, as I mentioned, we are using the guidelines from ACMG and AMP. And so the curators are using like a roadmap with these guidelines to assess the pathogenicity. And some of the curators are trained to use the ClinGen pathogenicity calculator. And because with this procedure, it's a bit more time consuming, but it's also highly robust, meaning that the curators come to the same conclusion, because it can be quite difficult to interpret variant impacts based on different information that we have. It's coming from predictions on the protein structure and function, but also functional information we have from literature. And then, of course, also animal models or mutagenesis assays. And yes, of course, also patient data that is described in the literature. So all this is used by the trained curators to use the ClinGen calculator, and then these assessments, they are also submitted to ClinVar.
All right. We have another question. Is it possible to use LC-MS mass spectrometry data to find proteomic variant? The service provider said they can give both raw and processed data.
I think, I'm not sure, but I think you can't just take a complete proteomic data set and feed that into UniProt and then use that to try to identify variants. I'm not sure, but I don't think that's possible. But we can investigate and answer.